And dear students of 12th class, and today in your botany, our topic is transcription unit and the gene, and we will try to learn about the proper definition of a gene, what a gene is. And now, if we go through the different definitions of the gene, ultimately we will look upon what your text or the NCERT text has said about the definition of the gene. Gene, a unit of heredity which is transferred from a parent to offspring and is held to determine some characteristic of offspring. Another definition, a distinct sequence of nucleotides forming part of chromosome, the order of which determines the order of monomers in a polypeptide or nucleic acid molecule which a cell may synthesize. Firstly, we must have understood where this gene lies. Basically, in all eukaryotes, the genes are located on the chromosomes. Chromosomes are held as the chromatin material in the nucleus of the eukaryotic cells, wherein the chromatin during its somatic phase, when it is as such until it reaches the cell reaches the divisional phase, wherein the chromatin gets aggregated into the solid and visible structures known as the chromosomes. The genes are located on these chromosomes, as we have already known that the genes or the sequence of nucleotides, a specific sequence of nucleotides in DNA or RNA that is located usually on a chromosome, and that is the functional unit of inheritance. Functional unit of inheritance means that it has a particular character to carry from parent to their offspring, and this controlling the transmission and expression of one or more traits by specifying the structure of a particular polypeptide and especially a protein, or controlling the function of the other genetic material. And now, what the NCERT book says about the gene. The gene is defined as the functional unit of inheritance. The functional unit of inheritance, though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence, because a particular DNA sequence is not formed only of the functional units of the gene. There are many other sequences that we are going to take up. The DNA sequence coding for transfer RNA or ribosomal RNA molecule also define a gene. So not only that part of the gene that codes for a particular protein or carries a particular or a specific character is the gene, but also the DNA sequence coding for the transfer RNA or ribosomal RNA molecule also define a gene. They are also the genes. However, by defining a cistron as a segment of DNA coding for a polypeptide. Now, cistron, a segment of DNA coding for a polypeptide, the structural gene in a transcriptional unit could be said as monocistronic. That means a structural gene may be comprised of just one cistron, as in most of the eukaryotes, or it may be polycistronic. That means in having many cistrons together, mostly in the bacteria or prokaryotes. And in eukaryotes, the monocistronic structural genes have interrupted coding sequences. The monocistronic structural genes have interrupted coding sequences. The genes in the eukaryotes are split. The genes in the eukaryotes are not continuous but are split. The coding sequences or expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. Exons are the sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in the mature or the processed RNA. So it is only the exons which are expressed, which appear in the mature or processed RNA, whereas the introns are the intervening sequences which do not appear in the mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of DNA segment. It means we can't say that gene is a segment of DNA because the split gene arrangement, it complicates the definition of a gene in terms of a DNA segment. So, inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. That means that a particular part of the DNA that is meant for carrying a particular character is also affected by the promoter and the regulatory sequences. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. So only a part from a particular DNA segment codes for RNA or the protein. Oh, thanks. Have a nice day.